So getting back to explaining the biochemistry of vitamin D. I want to emphasize the point that regardless of which way or what form of vitamin D we take in, the initial substrate is not the active form of vitamin D. To become fully activated, it needs to undergo two successive hydroxylations. Hydroxylation is an oxidative process facilitated by enzymes called hydroxylases that convert CH groups into COH groups. The first hydroxylation takes place in the liver on carbon 25 to form the prohormone 25-hydroxy-vitamin D, also known as calcidiol, but most commonly referred to as 25-OHD. The second hydroxylation takes place in the kidneys on carbon 1 to form the hormone 1,25-dihydroxy-vitamin D, also known as calcitriol, which is the fully activated form of vitamin D. Now, let's take a look at the biochemical mechanism by which calcitriol acts to improve calcium absorption in the small intestine. When calcium enters the lumen of the small intestine, it is brought into the intestinal cells, which are known as enterocytes, through a calcium transporter where it binds to proteins in the cytosol known as calbindin D. This allows the calcium to be transported to the other side of the enterocytes, where it can be pumped out into the bloodstream via an ATP-dependent calcium pump. This process is not very efficient, but when calcitriol is also present in the enterocytes, it binds to vitamin D receptors, actually entering the nuclei of these receptors, where it activates a genetic program which increases the translation, transcription, and ultimate expression of both the calcium transporter proteins and the calbindin D, resulting in increased activity of the calcium pumps. So in other words, when calcitriol is present, it increases the expression of specialized proteins within the enterocytes, which results in increased uptake of calcium into the enterocytes, as well as an increased uptake of calcium into the bloodstream, actually increasing absorption into the blood by a factor of 10. So, as you can see, it is this vitamin D-induced protein synthesis that is fundamental to how vitamin D's calcium absorption function is accomplished. Let's continue this discussion of vitamin D-induced synthesis of specialized proteins relating to another function, calcium utilization. This topic has already been covered in our vitamin K2 video, which I've linked below. So rather than repeat the whole discussion, I'll just highlight some of the main points. Vitamin D stimulates the body's synthesis of some other specialized proteins, which are characterized by their glutamic acid residues. These proteins, known as GLA proteins, have the unique ability to bind to calcium, but only when they have been activated by vitamin K. Because these GLA proteins depend on vitamin K for their activation, they are known as vitamin K-dependent proteins. Vitamin D is responsible for producing numerous GLA proteins, but in this presentation we'll only discuss two: matrix GLA protein and osteocalcin. Matrix GLA protein, also known as MGP, is a vitamin K-dependent protein produced by vitamin D and is secreted by arterial linings. When activated by vitamin K2, MGP supports the inhibition of calcification in the heart valves and arterial walls, and also supports the inhibition of kidney stone formation in the kidneys. Osteocalcin, also known as OC or BGLAP, is another vitamin K-dependent protein produced by vitamin D and is secreted by osteocytes in the bones. When activated by vitamin K2, OC shuttles calcium to the bone matrix in order to increase bone density. So both of these vitamin D-stimulated GLA proteins work in tandem with vitamin K2 in the utilization of calcium. Together, they work to shuttle calcium ions around in the body. The calcium ions are shuttled from where they should not be, like in the heart valves, arterial linings, and kidneys, to where they should be, the teeth and bones. Another function of vitamin D is to support the immune system, and as you can probably guess, as is the case with vitamin D's other functions, it relates to protein activity. It's also important to understand that vitamin D-induced protein is involved in modulating both innate and adaptive immune responses, both of which may be compromised under conditions of vitamin D insufficiency. Researchers have long observed that vitamin D deficiency is common amongst those with autoimmune disease. This link has prompted more studies, which has led researchers to discover that immune system cells are capable of synthesizing and responding to vitamin D and have the ability to convert calcidiol to calcitriol, which turns on key peptides, precursors of proteins, in the immune system. Mechanisms such as this are important in promoting antimicrobial response to pathogens in macrophages and are also important in regulating the maturation of antigen-presenting dendritic cells, which may be a key pathway by which vitamin D controls T cell function. Notably, the development of suppressor regulatory T cells, which are responsible for differentiating between outside invaders and self cells. Calcitriol essentially makes our immune system smarter, teaching it to not attack itself, supporting healthy immune responses and the suppression of autoimmune responses. Vitamin D also plays a role in anti-aging. Most of us know that our cells have a limited lifespan, and so they divide frequently in order to replace the ones that die. If our cells stopped dividing, we would die. The process of cell division is called mitosis. One of the steps of mitosis is the duplication of our chromosomes, which are in each cell's nucleus and contain our DNA, which is subdivided into our genes. The tip of each chromosome contains a protein coating referred to as a telomere, which protects the DNA, much like how the end of a shoelace protects the lace from fraying. Telomeres gradually get shorter over time, which is part of the normal aging process. A fascinating 2007 study published in Clinical Nutrition looked at a group of over 1,000 pairs of identical female twins between ages 18 and 79. The study demonstrated that the twins with lower vitamin D status had shorter telomeres than their twin counterpart who had a higher vitamin D status. The researchers concluded that their findings suggest vitamin D may have exhibited an anti-aging effect, and that this effect may be attributed to a combination of both vitamin D-induced protein synthesis and anti-inflammatory properties of vitamin D. It should be very clear from our discussion thus far that vitamin D's importance in supporting health goes far beyond the implications of its inverse correlation to rickets in children. Aside from numerous functions we've already discussed, adequate vitamin D is also known to be important for proper muscle function. Vitamin D inadequacy has been reported in approximately 36% of otherwise healthy young adults and up to 57% of general medicine inpatients in the United States. Even higher percentages have been reported in Europe. Recent epidemiological data have demonstrated there to be a high prevalence of vitamin D inadequacy among elderly patients, and especially among patients with osteoporosis. Researchers are now investigating the impact vitamin D deficiency could have on MS, diabetes, hypertension, and common cancers. In this presentation, I've spoken of vitamin D inadequacy, vitamin D insufficiency, and vitamin D deficiency. These terms are often confused with each other, so I will try to clarify their meanings, then segue into a discussion of recommended versus optimal vitamin D dosages. But in order to understand these topics, we need to first discuss the concept of vitamin D status. As stated previously, regardless of whether vitamin D is produced in the skin or ingested in the diet or taken as a supplement, it's biologically inert, requiring 2 successive hydroxylations to become biologically active. First in the liver to form calcidiol, then in the kidneys to form calcitriol. With the identification of calcidiol and calcitriol, analytical lab methods were developed to measure these vitamin D metabolites in the blood. You might think that because calcitriol is the active form of vitamin D, a measurement of its blood serum level would be a direct indication of vitamin D status. But actually, it turns out that calcitriol is not a good indicator of vitamin D status for 2 reasons. First of all, calcitriol has a very short half-life, which means that a low blood level could be more attributed to its short half-life than to a person's health status. Secondly, calcitriol synthesis is tightly regulated by the endocrine system using complex feedback loops involving parathyroid hormone, calcium, and phosphate. This results in calcitriol levels not typically decreasing until vitamin D deficiency is severe, making it not particularly useful as an indicator. Surprisingly, it's calcidiol, which we will refer to as 25-OHD from here on out, which turns out to be the best indicator for vitamin D status. 25-OHD blood serum levels are often expressed with units of either nanograms per milliliter or nanomoles per liter. In this presentation, we'll use the nanograms per milliliter convention, but the value can be converted to nanomoles per liter by multiplying it by 2.5. A 25-OHD blood serum level greater than 30 is considered a healthy level for all children and adults to maintain throughout the year. A level of less than 30 but more than 20 is considered vitamin D insufficiency or vitamin D inadequacy. A level of less than 20 is considered vitamin D deficiency. Okay, so now let's talk about vitamin D dosages, how they relate to vitamin D status, and why it's important to understand the difference between recommended dosages and optimal dosages. NHS Global has been producing DMAX, our vitamin D3 product, since 2003. DMAX is a 5,000 IU vitamin D3 product derived from lanolin and comes in a 250 count bottle. DMAX was formulated to be the perfect one-to-one complement to VK2, that's NHS Global's vitamin K2 product. DMAX can be taken by itself, but it's recommended to take one capsule of DMAX for each capsule of VK2. They should be taken together at the same time. For additional information, please visit vitamin-d-max.com or call toll-free 877-965-2140.